Hi, my name is Andrew Seaman, and I'm a sophomore in biomedical engineering here in WVU. I'm in my second semester of the RAP program, and I'm currently studying under Dr. Stan Hellman over in the Department of Physiology and Pharmacology. And I'm here to present to you a project that I've been helping out with this past semester that looks into the effects of chronic feed restriction on KNDy neuron expression in the ovine hypothalamus. This project is mainly concerned with the unknown neural connection between the metabolism and the endocrine system. As many of you know, the endocrine system is primarily made up of the hypothalamic pituitary gonadal axis, also known as the HPG axis. And this axis is directly controlled by the secretion of the decapeptide gonadotrophin releasing hormone, also known as GnRH. Further up in the hypothalamus, there are neurons that co-express the peptides kisspeptin, neurokinin B, and dynorphin, called KNDy neurons. And these neurons act as both a brake and a pedal for the secretion of GnRH. Specifically, both kisspeptin and NKB have a stimulatory effect on GnRH secretion, and the expression levels of these two peptides are directly related to the secretion of GnRH. It is also worth noting that the glycoprotein called luteinizing hormone, or LH, is a hormone secreted by the pituitary gland and can be used as an indirect way to monitor GnRH secretion through the blood. The premise of this project is to compare the differences in arcuate kisspeptin and NKB expressions in feed-restricted and normal-fed sheep, while also looking at LH secretion levels to draw conclusions on whether or not KISS and NKB are involved in the signaling pathway between the metabolic system and the endocrine system. So, to conduct our experiment, we separated our sheep into two groups, a normal-fed group and a restricted-fed group. The restricted-fed group were fed to lose around 15 to 20% of their body weight over a period of about 13 weeks. The sheep were bled every 2 weeks to get LH data in regards to time. Then the hypothalami were collected and stained for kisspeptin and NKB through the immunocytochemical process. An immunocytochemistry is a wet lab procedure that involves washing tissue samples over and over again in different solutions to wash away background material and highlight the desired proteins through immunofluorescence. And after all that, our tissues were digitally scanned onto a computer using a scanning microscope, and neurons were individually counted using computer software. And in figure 3 in the top right-hand corner is what the neurons look like on my computer screen and how I was counting them and what the neurons look like. So, as of now, our group is still gathering and processing data, so no results are finalized. However, from our collected data, we've been able to see a clear difference in weight between the feed-restricted and the normal-fed sheep, and the LH secretion is considerably lower in feed-restricted sheep. And figure 1 represents the weight difference between the feed-restricted and our normal-fed sheep. And in figure 2, on top we have a graph of the LH secretion levels of the normal-fed sheep. In the bottom graph is the LH secretion of the feed-restricted sheep. As for neuron expressions, we have many more tissue samples to count, but our data at the moment is showing a slight decrease in arcuate kisspeptin and NKB expression in the feed-restricted sheep, as shown in figure 4. What we can conclude so far in this experiment is that our target weight for the feed-restricted sheep was met, and we did see an expected decrease in LH secretion. And although we can see a slight decrease in KISS and NKB expression in the feed-restricted group, the decrease is not large enough to be considered significant. We also do not have enough tissue samples counted to provide evidence that this slight decrease is a trend. In finishing the counting of our tissue samples we have collected, plus more testing on additional sheep and possibly mice, are necessary before we could draw any solid conclusions from this experiment. My name is Andrew Seaman. That's about all I got. Don't be afraid to ask any questions. Thank you for watching.